Hello, everyone. My name's Clay Moore. I know my title is very long, but it gives a good scope of what we're trying to do. I'm a drug discovery scientist at Rare Earth Genomics. I'm also a PhD student at Texas A&M University, and I'm co-founder of a new company called Novel Therapeutics. I've attended CannMed for the past 3 years now. It is a pleasure and an honor to be in front of all of you today here in Puerto Rico. I've made many great connections here over the years, such as Medicinal Genomics and Advanced Nutrients, who supplies nutrients to Texas A&M University for all of our research projects and for all of our plants. I also see a lot of familiar faces from other conferences, and noticing that it's your first time here, and it's very great to see all of you. My topic today is related to my current work and research, discovering a new chemotherapy drug to treat adenocarcinoma lung cancer with phytochemicals, artificial intelligence, and high-throughput technology. Cancer and neurodegeneration drive over 60% of the global mortality, yet the vast chemical space of plant-derived molecules remains untapped for modern drug discovery. We have a vast amount of peer-reviewed journals, books, and testimony from physicians, nurses, biologists, chemists, computational scientists, the list goes on, for hundreds, if not thousands, of diseases treated with some form of plant-derived medicine. Yet the current pharmaceutical industry continuously tries to improve fully synthetic molecules from scratch that usually do more harm than good. For over 400 million years, these plants have been evolving to produce more and more phytochemical diversity. For thousands of years, humans have made extracts from plants to treat various diseases, even all the way back to 2700 BCE. Zhenrong from China would make a tea extract with various plants, including hemp, to treat symptoms such as gout, malaria, and pain. Now, in modern day, we have access to technologies that can see, think, predict, and analyze better than 92% of humans when diagnosing, treating, and inventing new methods that we have never thought of previously. And so today I'm going to give you a technical overview of an artificial intelligence that I built for drug discovery and give you the core concepts that are important to take home. So the program starts out with a SMILE string that is pulled from a database that I curated over 200,000 known phytochemicals. This is cannabidivarin, or CBDV. A SMILE string is a line of code that is a 2D molecular descriptor which allows the AI to know exactly how the molecule it is starting with. It is comprised of elements and parentheses, which tells the AI which order a carbon chain goes, which way the molecule is oriented, where a double bond is, and so on. Now, a lot of us are more familiar with this. This is how we interpret molecules from a 2D perspective. The program then makes thousands of modifications, starting SMILE strings in parallel. As it is modifying, it is also filtering for molecules that allow for the laws of physics, has drug-likeness properties, predicted absorption, distribution, metabolism, excretion, toxicity, and so on. And once the program has gone through all of the modification that is set in the AI's parameters, it translates the 2D descriptor into a 3D dockable model. Now, this is important if we're trying to target receptors for cancers and neurological disorders. For instance, here's a 3D model of the HER2 protein pathway. HER2 is the most overexpressed pathway on the surface of most cancer cells. This is what the AI sees before it tries to dock the modified molecule for binding affinity and other molecular dynamic properties. For the respect of our human eyes, we can give it a surface. In the program, it is docking every single one of those modified molecules to all of the receptors that are set in the parameters that are in the database to receive a binding affinity score. If we zoom in, we can also see how the molecule is pocketed. Now, binding affinity is important, but there is way more to consider before moving on with a computer-designed drug. We have various tools to help us predict molecular properties for the medicinal chemistry, tools to predict off-targeting. As you can see, we screened the HER2 pathway, but it noticed that it would likely affect CB1 and CB2 as well because it's CBDV. We also try to predict ADMET scores that take all previous pharmacological knowledge to help to keep human health #1 priority. Now, it's very exciting to see this promising data on a computer for a modified molecule. But how are we going to make this molecule? Luckily, there are people a lot smarter than I that have developed an artificial intelligence for retrosynthesis. This is the stage that I am currently at in my PhD. I take the curated phytochemical database plus a modified phytochemical and their database of over 17 million known molecules on the planet Earth, and is then run through their AI to predict the instructions for developing the molecule in a wet lab. There are 3 ways that it can predict. The most common way is chemistry reactions, where it will lay out the entire tree step for step from the original phytochemical that it picked from. And then the second way is biochemistry, which can be produced enzymatically through a yeast or a bacteria model. And lastly, biology, which can be very difficult to control, but for example, how delta-9 tetrahydrocannabinol transitions into 11-hydroxy tetrahydrocannabinol when ingested orally. Through trial and error in the wet lab, the modified molecule is produced, then validated with analytical equipment such as gas chromatography, liquid chromatography, and near-infrared, and so on. Once we have selected our modified molecule from the AI and developed it in the wet lab, we can then move into high-throughput in vitro screening. First, we prepare cell lines, such as a cancer cell or a neuron cell in an incubator, and we can then add them into a large automated system for screening a molecule across the cells in various dosages. We currently have the power and precision to do up to 1,536 well microplates, whereas most dosing is done at 396 well plates or less. We then wait for a period of time, allowing the molecule to interact with the cell line. We then put the cells in a machine that can produce a high-resolution image of what is happening, or what mechanism is occurring at the cellular level, whether it is apoptosis, programmed cell death, tumor suppression, viability, and so on. It's good to note that we also run formulations of multiple phytochemicals and modified molecules across cell lines for synergistic effects or an entourage effect. It's good to note that we can take this data and decide to move forward with it into in vivo mouse models, speak with the FDA, partner with the CRO, and so on. Either way, we can feed all of this data back into the AI to teach it to make better decisions in future drug discovery. Just to put it into perspective, we can feed all of the computational modeling, phytochemical databases, and calculations back into the AI. We can input all of the data we receive from real-life experiments, from extraction to synthesis to in vitro and in vivo mouse models back into the AI. And ultimately, this is to build a tool to help develop safer and more effective treatments than current market leaders. Now, AI discovery has made great strides in just the past couple of years. There's been 67 AI-designed drug candidates in clinical trials right now. There's 21 candidates that have already cleared phase 1, and there are 7 IND clearances for fully AI-designed molecules that have no cell testing. 30% of new drugs expected to be AI discovered by 2025. This year, 30% of all new drugs are going to be developed by artificial intelligence, and that number will only increase over the years to come. FDA momentum, there's more than 500 submissions containing AI compounds reviewed since 2016. And so what this means for us is that it can align us with the 2025 FDA draft guidance to build IND-ready data packages from day 1, exploit higher early phase succession to shorten timelines and lower risk, target IND filings Q4 2026, post-adeno carcinoma screen and GLP toxicity, leverage high-throughput technology, rapid generation of pre-FDA efficacy and safety data. So this is very important because right now, to develop a drug in the known industry, it takes over $2 billion per drug, and there's only a 10% success rate over all of these drugs. 90% fail. But other than that, thank you guys for listening to my talk. You can follow up with me in the hallway, or you can connect with me via email or my LinkedIn in the QR code. I appreciate your time today. Thank you all very much.